Hi, wonderful. So welcome to our webinar series, The Rise of Live Cell Shipment. My name is Kathrin Adlkofer and I'm very happy to have two speaker or discussion partner. I have Johana Kuncová-Kallio, Director of UPM Biomedicals at UPM, a company who is the leader in bio-based products for biomedical application and healthcare. So hello. I have also Lauri Pasonnen, who is, actually you have to introduce yourself. He's helping the reference in a specific question regarding our topic today, also from UPM, and I'm very happy that you're both here. But before I start asking questions and hoping to learn a lot about your company and your products, I would like to introduce to you two things. One is that in this webinar series, because what we want to actually grow a community of live cell traveler, so that cells are traveling and you have the chance to win such a wonderful small box. I hold it. You know, it's a Cellbox in small. And if you bring a friend, you get one. And if you have 10 of these, we will actually send you one of our cell bags where you can also transport cells within. So this is a little idea of creating a community of people who are interested of how we can transport cells safe and sound without freezing if there is freezing, if there's no opportunity to do so. Second, I would like to introduce you a little bit to the topic before we start with our discussion partner about the challenge and the cultivating of transporting fragile cells and tissues. And I know that some of you might have already seen this, the slides, but I have a couple of new ones as well. So giving the idea that we have, years ago, cell biologists were working actually with very simple cell systems, cells which you can easily freeze and thaw, and the cells do not change, or probably they already changed, but it was not that important. Throughout the development of, like the cell biology and the learning, we created more and more cells which have, like the biological behind the cells are much more informative. So we start reprogramming cells. We have primary cell lines which are very fragile and which are really hard to freeze down and to thaw them again. And then of course, the cells are even getting more complex when you talk about organoid structures, 3D tissues which are getting printed, cell models, and there actually we are in a situation where you actually even can't freeze them. So how do you actually transport them? And when you look at the globalization within the drug discovery, within the development, as well as in the field of research, you can see people work all over the world very close together. And how can you transport these kind of complex cell systems or fragile cells if you can't freeze them? And this is actually the topic we are challenging. And even further, when you look a little bit more detailed into the drug discovery development process, you can see throughout the whole pipeline that there are many application opportunities where you have to work on the process, how to optimize the logistic, so that you can use the cells which you process on another spot, in another area, in another country, having the same quality and the same quantity. And this is actually why we are, starting, or why we start up our Cellbox. But of course, not only Cellbox, but also other applications are needed to stabilize this process of logistic. And I'm very happy to have my discussion partner here today, because UPM has, products which are really helping stabilizing cells when you transport them, when you keep them in culture, to keep them safe and sound. And Johana, I would be very happy if you would introduce yourself. And of course, Lauri, that would be, if you could also say a word, and then maybe give us a little introduction about your company.
Sure. Thank you very much, Kathrin, for the introduction. Actually, my name is Johana Kuncová-Kallio, and as was said, I'm the director of UPM Biomedicals. Lauri Pasonnen is here with me. He's the senior application scientist, and he's the guy who actually knows all the cells quite, I would say, he's familiar with each cell type that has been going through our gels. And actually, my first experiences with transportation of cells is 20 years ago, traveling from one university to another. One of us was driving and the other one was on the other seat holding a box of styrofoam with petri dishes in it filled with cells and Leibovitz medium, and kind of acting as a gyroscope and a life incubator. And I think that this is something which we have soon noticed, that if you take the cells, the time starts ticking, and you want to have the same conditions for the cells throughout the whole way until you start the experiment. Otherwise you get into trouble. But getting a little bit into the presentation, I hope you can see my slides. Can you confirm?
Yeah.
Good.
Wonderful.
So
I can.
Perfect. So in fact, where we come from as UPM, UPM is a over 100 years old company, which has been famous for forest industry, and in the recent years ventures into new areas, and one of those areas is biomedicals. And we have not selected any of the easy topics. We have gone all the way to areas where we are in the clinical applications and also dealing with the most precious cells like primary stem cells. Why a forest company would be going into biomedical is shown here, because we start from the tree. In this case, we have birch. We go through the chips and cellulose, where normally the process would be really stopping in terms of refining. But now we go one step further. We go into nanocellulose, and this is the core material for all of our products at the moment. The nanometer size is around 100,000th part of a paper sheet thickness. So that's just to give you an idea. And as you know, cells are around few micrometers, so it's a perfect environment for them to live in. We have heavily invested. You can actually meet Lauri here, and he's there answering your questions if you have any. We have actually generated a quite large portfolio in this area. We have more than 300 patents around nanofibrillar cellulose, which is the core of this products. And also we have more than 150 optimized protocols for cell cultivation, and especially looking into the 3D cell culture, which we are all about. Now going into, let me see, into the products. We have actually all of our products, as I said, is based on the gel. And for the life sciences, we have GrowDex, which is material used for preservation of the 3D matrix of the cells. But also we have 3D printing of cells and gels together, so GrowInk series. And then we have our first clinical product. So what is good to say is that we are manufacturing for most of the part under the ISO standardization. And since we have a clinical product, we also hope that with these gels, we will be able to access the most crucial applications in regenerative medicine in the future. Our view in these areas is pretty much in line with what you have, Kathrin, described. And the idea is that we are kind of with the GrowDex starting already ideal in the pathologist area, where we try to kind of stop aging the cells. I would not say that we can, make them young again, but we can preserve the biomarkers until they get into the processing. And in this case, now the newest trend is, of course, to get them into the 3D culture, whether it's organoid or tumoroid or spheroid. But that is now the crucial part, how to get them into the place where they can be used, and then later we are able to remove the gel and get them sequenced, which is also what people want to see, the functionality of the cells afterwards, not just what happens in the high throughput screening. But also we are working on topics related to personalized medicine and looking at the patient-derived explants, and we are also very much into regenerative medicine, which is the far away goal still, but we are working in the 3D printing, we are looking at the cell release and also freeze drying of the tissues and structures. I have a short video just emphasizing the, let me see if I'm able to get it running. This is the so-called demo effect. Just to kind of show the idea that we are not just developing any gel, but we are really focusing on the natural sources. So we are here working with birch and the renewable material as such. So no animal DNA, which would be interfering with the results. So I let the trees and the finished forest to sink. And as you can see, we are also able to easily pipette them and also easily dispense, obviously. And this is for us. So overall, we have this range of products, the GrowDex, which has been the longest on the market, the GrowDex T, which is a newer kit on the block and which is especially useful for the neural cultures. And then we have GrowDex A, which is the newest one, which is a matrix that can be functionalized via the avidin biotin linking. And also we have the GrowDays, which is an enzyme that allows us to take away all of the gel and leave the cells intact for the further action. Again, coming back to why this gel and not something else, the most important is really that it's animal-free. There is no animal DNA. It's just birch and water. No surprises in the sequences. Since there is no animal origin, it's also easy to keep the variation down, which means that when you get results from your assay, it's the variation comes from the drugs that you introduced, not from the gel that you preserve your cells in. We are able to remove the cells from the gel. We are able to screen them with fluorescence, which is again a big benefit when you are talking about the pharma applications. We can tune the viscosity depending on the cell type and what the people in lab love, that you can work with the material at room temperature. You don't have to keep your hands cold. So that's pretty much it. And this is my contact. If anyone is interested, I would like to remind once more that the Lauri is there in the chat waiting for your questions. If you have any specific questions, please do ask. And I'm looking forward now, Kathrin, to your questions.
I have one question before I go into detail. Why birch? Why did you do? Why is it birch?
Actually, there has been some 20 years ago research into many sources of nanocellulose, and in fact, this was the one that had the best properties for cell cultivation. So there has been many other directions. There has been, paper packaging for food. There has been, additives for paints, and with UPM, the direction has always been that we are, one of the largest companies, and we can afford to take the most tough application, which is the medical part, and which means that really the cells and the toxicology were the first things that came in, and then the birch came as the best.
So birch, it is birch. You mentioned that, especially that GrowDex is particularly effective in neurons and neuronal organoids. And can you refer to that in a little bit more detail? So why does these cell types do especially well in GrowDex?
Yes. So in fact, we have many cell types that do well in GrowDex, but the neural cells are such that they prefer actually the GrowDex T, which is the newer version of GrowDex. And that GrowDex-T, here you can see a nice example from Jan Bruder from Max Planck Institute. That GrowDex-T is an anionic version of the nanofibrillated cellulose, while the standard GrowDex is just native cellulose. And we believe that one of the things that the neural cells like is the charge, but it may be also that it's related more to the structure of the fibers within the matrix. This is something we can only guess. We don't know. The cells don't answer. We try to ask, but we try to ask with the researchers and get more answers there. But this is one of the examples.
Okay, go ahead. Sorry.
Just wanted to show this example with the midbrain neural organoids, which I think is one of the top examples we have in this field, where again, this has been growing in GrowDex-T for 22 days. That's pretty impressive for neural cells, I would say.
Okay. So do you think it's because of the structure? So you believe, I mean, it must
or
be
charge.
Oh, the charge. Yeah. Okay.
Yeah. So it's the anionic surface of the nanocellulose is maybe the other thing, but again, it's very hard to prove which is the actual final reasoning why. But we see definitely better results with neural cells in GrowDex-T than with the regular GrowDex.
What about primary cardiomyocytes, or like cardiomyocytes? Did you see any effect on that?
I mean, with the cardiomyocytes, you can grow them in both the GrowDex and GrowDex-T. We will have protocols you will find in both. But I would say that for some reason the neural cells are more, they are picky. They are tough guys.
But they're also, I mean, extremely fragile. Especially if you want to keep them adherent. So I also wondered why. If you have this plant-based material within your cell culture, and having all these polysaccharides and cellulose, which is probably affecting the metabolism. So is there something? Is there an effect, or because I want to keep the cells exactly in the native environment? If you add something like cellulose, does that change the environment, or do you see any effect on the metabolism?
So in fact, what we have done so far, so in GrowDex, obviously we are looking at the sequencing and we are looking at the activity of the biomarkers. And then when we are working with GrowDase, which is the enzyme which we use to remove the gel, actually it's the GrowDase removes the gel or reduces it to glucose. And in fact, this is some of the examples showing the cell structure after they have been in GrowDex, and then the GrowDex has been removed by GrowDase, and we can see that the structure of this HepG2 spheroids looks pretty good. And then we have also the functionality shown here with the IPS spheroids after 12 days culture in the GrowDex. Of course, there would be other techniques you can use to characterize how well you can preserve the functionality and how well you can preserve the morphology. But this is something where I would say we have so many publications already that I would say it's shown that we don't have any detrimental effect on the cells. On contrary.
Yeah, because this is what I learned. When you want to keep the cells in a state not affected from the environment at all. Even, I mean, it's same when you freeze them down and you put DMSO. That definitely will some kind of affect the cells. And of course the question, like if you suddenly put a gel in it, that the cells might be, and even the grow base might affect, and if you have the data shown that this is actually not affecting, that is very important for the scientists.
I think what is more important in terms of kind of causing side effects or side results of the unwanted artifacts in the results, I think is again that it's just virgin water. If you consider what are the other options where you have animal derivatives and you actually you could compare it that it's like having the cells on steroids. In our case, they are really in like a clean environment, which is well defined, and we don't have any unknown parts in it. And also in terms of DNA, again, no animal DNA. It keeps it pure.
Yeah. You mentioned that you have two products. One is the Grodex and one is Grodex T.
Yes.
So can you explain a little bit the difference? So when do I have to use which products?
Yeah, so in fact, I think that the best difference is one of them is the neural cells. So neural cells for whether it's the structure or whether it's the charge, they prefer the Grodex T, which is the anionic version of the nanofibrillary cellulose. The second difference comes from imaging. So this is a patient-derived prostate cancer tumoroid, and you can see this is 10 times and 20 times magnification. And this is the same kind of experiment with Grodex, and this is in Grodex T. And I would say that in the 10 times and 20 times, there is not much difference that would be visible. But when you go to 40 times, you will see the difference a little bit more. So let's say if you are planning to have screens where you are planning to use 40x magnification, and especially where you are planning to use phase contrast, I think then I would go for Grodex T. On the other hand, in most of the other applications, if you are just going to use fluorescence, there is not going to be much difference, as you can see.
Okay, are you going to keep on, having specialized Grodex for different cell types? Do you keep on developing these kind of, so that I have to use this one for neurons and this one for whatever CAR cells? Is that a direction you would like to look at?
I think that these two kind of basic versions are something that you can customize, and this is something which, I mean, we cannot cover every cell in the world. See, the idea is that then we have the Grodex A, which is allowing you to kind of tailor the surface to your needs. So if you want to have specific capture for, let's say, stem cells in certain differentiation stage, you can do that by modifying the Grodex with a specific capture, and then let the cells that get anchored to it, they are the ones you're interested in. Or you can have, we have actually very nice results showing whether you have the growth factors in the matrix attached or whether you are freely floating in the media. We have been able to show big differences in the, so there is a big benefit if you tailor the matrix a little bit further. But we think that in that case, it's better that the researchers themselves have the choice and have the capability to kind of modify themselves. Then, in most of the cases, they are the experts on that particular cell type.
One more question regarding the viscosity of, so can you change that so that they're really viscous or that it's more gel-like or almost fluid? So is there a difference? Or can you use it in different liquidity stages? I don't know if you
so
say
both of the gels have tunable viscosity. So it's just depending on how much you dilute them with the media. So in a way, the concentration can be varied. The Grodex T by default is a little bit more stiff, but again, you can dilute it to more fluent version. I think that in both of the Grodex versions, you have to search for the right stiffness for the different cell type. So again, you know that the cells which are that are ready to experience higher shear forces or cells that are exposed to more in vivo-like conditions, you want to have different stiffness for different cell types. So you can test different stiffnesses and then see which of them suits your cells the best.
Like when I imagine what kind of disposable actually is used. I mean, I could see microtiter plates, the big ones, especially when you have them and they are, I mean, if you have normal media in it, it's really hard to transport them, you know, even from one refrigerator to the other, or incubator to the other. So, you know, what is the common disposable you are actually working with, or your customers using your product?
I would say that this would be a good question for Lauri because he has much more experiences. I would say that the well plates, but Lauri, please.
Yes, very good question. Thanks, Johana and Kathrin. So 96 well plate is the most commonly used well plate format with Grodex. So basically the wells are in good size that you can apply, for example, 100 microliters of the hydrogel with cells to the well, and then, for example, the same volume of the cell culture medium on top of the hydrogel. And then you can replace the medium whenever you need. Also, smaller wells, the 384 well plates, have been used especially in the, let's say, more higher throughput applications, where you can use also automated dispensers.
One question. I mean, if they're really, you know, like the 384, they are really small, and if you pipe, I mean, if you dispense them, you can actually easily transport without any, you know, because it's too small, and the viscosity of the liquid itself is already good enough. I understand 96 well, because, you know, when you shake them, they spill over. But the very small one, you know, so why would someone needs to? Is it for long distance traveling then, or why would you need that?
I'm referring to the normal 3D cell culture protocols, where, for example, high throughput screening units, they are screening different drug molecules, and of course they want to increase the throughput by using the smaller wells. But you're absolutely correct that in smaller well you have more of the wall support to also support the cells inside the hydrogel. But yes, Grodex gives the physical protection, physical support for the cells in basically any type of a well.
And the most common, you said, is the 96 well so far, which you're using. Yeah. Okay.
Yes.
And so what kind of, so you probably already transported cells in Grodex. Can you explain us a little bit, so the experience you made, and also the challenge, not only that it works perfectly all right, but also the challenge, and actually what you also believe, maybe both of you, were the development of the market, you know, because I mean, I really believe that shipping live cells or cells alive will be actually the future transportation of cells. And maybe I would like to hear your opinion, both of you.
I will maybe split it into two questions. So I will maybe speak about more of the future, and I would ask then Lauri to speak about more of the experiences and his view of the future. So I will for a while show this, and in fact what I think is that the transportation of live cells is going to increase as the value of the cells that you're transporting is increasing. So let's say that if you are transporting you know, standard cell lines, it's not likely worth it. But when you have added value like 3D organoids or tumoroids, if you have organoid chip or if you have 3D printed products, there you have put already so much added value that the freezing is too high of a risk to take. My view is that actually the transportation in an incubator conditions is something that will enable new types of companies altogether. So if you think of the process now, the 3D cultures, if you're not able to ship them, or you have like 50-50 chances that your organoids are still alive on the other side when you thaw them, it means that either the pharma companies have to produce the organoids themselves, which is expensive because it means you have to tie a lot of stuff and equipment to that. The other option is that they use service companies where they ship their precious drugs and get them screened, which is not always a choice they want to make. So I think that in the future there will be more and more companies that will be providing the service of, developing the 3D organs, mini organs, tumoroids, and have them ready to ship for, the screens that the pharma orders. So then they will order that, okay, we want 100 mini hearts. Can you deliver them to us next week? And then you get them cultured and ready. So I expect that within the next years, we will see more and more companies that will be selling ready sets of 3D cultured cells, organ on chips, or 3D printed products.
Yeah.
And now I give the word to Lauri as well.
Thanks. So yeah, about experience of transportation of different, let's say, biological materials, we already have quite a few of examples. Of course, the first starting from the easiest way, the cell lines, robust cell lines can be transported in GrowDex. That has been tested. But the example that Johana showed with the Jan Bruder example with the neural organoids, I would say that this is very good example, as we know that the organoids, they are already more sensitive and more complex cell structures. And to be able to, yes, thank you. So to be able to also demonstrate the activity, the viability, the functionality of these organoids is crucial when you are thinking about transportation of the more complex biological systems like these organoids. So this is a very good experience, very good example, like a success story, how GrowDex can help in the transportation. But in addition to the cell lines and this neuronal organoid example, we have, of course, experience with, for example, different materials like bacteriophages. So the small viruses that kill only bacteria that could be used for treatment of antibiotic-resistant infections. And we have shipped those bacteriophages in GrowDex in winter time in Finland when we have quite cold weather. And the phages, they remain viable, and it seems that also for that kind of a biological material, GrowDex is very good transportation material. So we have already few examples, but of course, for example, the transportation of biopsies and tissue pieces, that is still ongoing, and we have some collaboration projects ongoing.
One question I'm thinking, so biobanking, this is really something which is, it's actually dramatic to see how different the way tissue gets retrieved from the patient and then brought to biobanks, So actually they all arrive in a completely different condition and stages. And to standardize it, always keep them in the same temperature, always keep them in the same media, maybe sterilized so that they cannot harm when you shake them. Look at tissue when you transport it for a longer distance, how it changes the structure in liquids. Do you think there is a market if you would come up with the idea of standardizing biobank material using GrowDex as a standard transportation media? Probably already could help,
Actually, we have quite a few projects. We have something like 60 projects ongoing in different topics around the life sciences and clinical applications. And actually, we have already now experiences from the pathology when working towards the patient explants. So at this point, each of the samples, when they arrive from the surgery, are being processed first in the pathologist, who is then part sends to biobank, part goes maybe for research, part goes maybe for some other testing. And when that piece arrives, you have to work it through immediately because it comes just in the media and it kind of the clock starts ticking immediately. There is no waiting. While when we are able to ship those in GrowDex, it means that we are able a little bit slow down the aging, I would say in layman terms, and kind of being able to wait for the other samples to arrive from different surgeries and then process them all at the same time and have kind of the luxury of not being, it makes it much more cost efficient when you're able to do it all at once. And for the GrowDex, in this case, we have had excellent success with the explants. So we are hoping that there we will also find more. But in this personalized medicine and in the regenerative medicine, they are very young areas, and we hope that they will be flourishing, of course. But at this point, I think that the biggest challenge in personalized medicine is to figure out who will eventually be the customer. Is it the local hospital that will decide on the treatment plan, or is it maybe the patient himself that he can have this luxury of choice, or is it the pharma company that will pay the bill that their drug will get tested, that it's the best solution for the patient?
I have one question. Did you ever look in the IVF market?
A little bit.
In vitro fertilization. I mean, they are really, it's such a fragile situation, where the cells are fertilized and keep them stable. I mean, I'm not that familiar. I only know that also their transportation is very important, and even when you walk them over the, like from one room to the other, it is already, dangerous.
I think for us, the ultimate goal would be really that we would be there from the beginning all the way until you return the cells back to the patient, in terms of whether it's IVF or regenerative medicine. But also we have to understand that in many of these cases, the researchers are the forerunners and the regulatory is not ready yet. Yes. So in fact, in most of the cases in regenerative medicine, we would love to already, we have a lot of researchers working with stem cells, and they are asking us, so when can we go to the clinics? And saying, you have to hold your horses for a little bit, because the regulatory offices need to provide more guideline as how these advanced therapies are going to be regulated. Yes. So we are now kind of waiting, in the waiting mode and hoping mode.
Basically, I hope that the whole regulatory aspects, they are also really hindering, innovation to get through to the patient or becoming a product. But this is a different story. One question I have. So if you say, what is the best product in the moment? So what are the best cells or the where you use your product most? Do you have any?
I would say, I'll send this question to Lauri again. He knows most of our customers.
Yeah, well, I would say that our customers, definitely oncology is one of the main application areas. So producing more relevant, more in vivo relevant, more physiological relevant in vitro models with cancer cells. But that's probably the reason that so much cancer research is done all around the world, and also much more better and more relevant in vitro models are needed. So there's a lot of interest in that field. But in addition to the cancer research, I would say also the liver cells, to make liver models 3D to test, for example, the hepatotoxicity of new drug compounds. And then, of course, stem cell and regenerative medicine is also one big application area. So if I had to choose three, I would say these three. Okay,
thank you. Interesting. Last little question, and then I have to close up. So are they able to, cells migrate? They know how to, they go a direction, keep in contact. Can they migrate through the matrix actually, or are they stiff and they are not able to migrate? Lauri.
So this feeds back to the tuning of the matrix for the needs of the cells. And I will throw this back to Lauri again, who has the most experience on this.
Yes, this is a very good question and good point. So the nanofibers in GrowDex hydrogels, they are not cross-linked. So it's not that stiff hydrogel that would prevent the cells migrating. So we have some live cell imaging videos where we can see, for example, that I think it was with the human dermal fibroblasts, that they can migrate, they find each other, and they start forming the spheroid structures inside the hydrogel. But as Johana mentioned, this can be, of course, adjusted by adjusting the stiffness of the material. But also with the higher stiffnesses, we can see that the cells are not completely jammed in the one place, but they can push the fibers around them, they can migrate, and they can proliferate also to grow larger spheroids.
Yeah, because I think this is very important. Yeah, thank you so much. Actually, we only have 4 more minutes, and for me, maybe to close that up, I think you know, when we started Cellbox, and actually our first box, there was even an adapter for the 10 centimeter dishes, and I was wondering, how do you want to transport a 10 centimeter dish with media in it from A to B without spilling the whole media? One way could be using your technique, you know, one is holding it, and so that was actually something which we also got asked from customers, how do you do that? And actually 96 well plates, or if you have flasks or tubes or bags, that works, but there are also applications where it would be really terrific if you could keep it more viscous, you know, and therefore, especially in the adherent cells, you know, that they don't detach. We did a lot of trials regarding how can we do that, and when we found you, or you found us, I think it's really helpful for our Cellbox using this kind of material, and I think we both know that this will be the right direction of you know, keeping cells you know, happy even when they have to be transported from A to B. And I hope that we stay in contact. I know that we will, but we will have other topics. The next time we will have another webinar in October the 7th, again at 4 o'clock, like always, so that even the US people can join us. And it's about the importance of logistic for organ on a chip. So organ on a chip, you know, like they can't be transported in the moment. So if someone is working with organ on a chip, they have to do it, and they have to do in-house services and not transporting these fragile systems throughout the country, or even to another country. And that's why we are very happy to get tissues from Berlin, and they are introducing us to the speaker. My understanding will be Uwe Marx, or any other senior scientists who are specialized in that field. And yeah, I hope that you might join us. I hope that you stay healthy, Johana and Lauri, and that we should keep in contact. And I say thank you to everybody. You know, listen to our webinar, and hope to see you all soon next time.
Thank you, Kathrin, for your kind words. Thank you. Goodbye.
And good luck to both of you. Okay. Bye bye.
Thank you.
Bye.
You too.